Frontier Flash, Future News. In December 2023, the U.S. Food and Drug Administration approved the first CRISPR-based gene editing therapy for sickle cell disease. The therapy, developed by Vertex Pharmaceuticals and CRISPR Therapeutics, marks the first time a CRISPR treatment has received regulatory approval in the United States. This is not a research milestone. It is a clinical one. Default tech sickle cell disease is caused by a single mutation in the gene that codes for hemoglobin. This mutation causes red blood cells to become rigid and crescent-shaped, leading to severe pain, organ damage, and reduced life expectancy. The approved therapy does not directly repair the mutation. Instead, it edits a patient's own hematopoietic stem cells outside the body. Using CRISPR-Cas9, the therapy disables a regulatory gene that suppresses fetal hemoglobin production. Fetal hemoglobin does not sickle. By reactivating fetal hemoglobin production, the therapy allows red blood cells to function normally, reducing or eliminating painful crises. Clinical trials showed that the majority of treated patients were free from severe vaso-occlusive episodes for at least a year after treatment. However, the process is intensive. Patients must undergo chemotherapy to clear existing bone marrow before reinfusion of edited cells. This is not a simple outpatient injection. It is a complex medical procedure. This approval signals three structural shifts. First, gene editing has moved from laboratory promise to regulated medicine. Second, the regulatory system is now willing to approve CRISPR-based interventions, which lowers uncertainty for future therapies. Third, this validates a business model for programmable medicine. However, there are constraints. The therapy is expensive. Estimates place the cost above $2 million per patient. The treatment process requires specialized centers and carries risks associated with chemotherapy. This is not yet scalable global medicine, but it establishes precedent. The deeper signal is this: biology is becoming programmable in clinical settings. This approval does not mean gene editing is routine, but it demonstrates that precision genomic intervention can meet regulatory standards. If future therapies reduce cost, risk, and complexity, this could mark the beginning of a new class of medicine. The frontier is entering the clinic.